Hello friends, this is from Chambers and today we'll be talking about the next chapter of histology, our histology classroom, that is histology of skin. Now histology of skin is important from the dermatology point of view. So I'll be talking the different microanatomical structures present in the skin, the various layers, and will be giving you a little bit of clinical correlation with the diseases associated with it. Okay? So let us share the screen. Okay, friends. So right now on your screen are the microscopic images of the skin section. Okay. Now this way, the section has been taken in such a way that this is the, if I talk about this right one, this is the outside. Okay, outer region, outer. Okay, and you are going towards the center. Okay, this is the periphery. And if I tilt this like this, so you can see it's the same periphery. First we need orientation, isn't it? So periphery, and this is the inside. Okay, coming towards the we're going downwards towards the layers, and this is the same diagram. So first thing I want to tell you, the basic concept which you should understand is you should know what are the layers. What are the layers? How to remember? The first layer you would be tackling, so the first layer which we will be tackling is stratum corneum. Okay. First you should write like this. You should write the skin, human skin. You know the skin is the largest organ of your body, isn't it? It is the largest organ of your body. It is having three layers. It is having three layers. Epidermis, dermis, subcutaneous fat. That is called panniculus. Okay. Now epidermis epidermis is made up of, okay, you can also write it the subcutaneous fat, you can also write what, hypodermis or subcutis. Okay, hypodermis or subcutis. So what is in the epidermis is, the epidermis contains what is made up of a clear stratified superficial, should write it, it is a superficial. Okay, and it is stratified squamous epithelium. Okay, stratified squamous epithelium. Understood? So if I draw something, if this is, let us remove this. Okay. Now this epidermis, this is all about epidermis. Okay, hit this point. Let me show you first, make you understand what these diagrams are. Where we need to first have a broad outlook overview of the sea. This is just the general overview. The magnification is not much. So from this side till here to here, this is all epidermis. This is all epidermis. Okay, this all is epidermis. Clear? It is shown also here. So till here is the dermis. Till here is the dermis. Okay, and the more you go inside towards, from the dermis to inside, is hypodermis, or you said subcutaneous fat. Can you see the fat lobules from the histology? Can we see the fat lobules, adipocytes? Can we see some septations also, fibrous septations? We can see some fibrous septation. So hypodermis is, that's why I wrote subcutaneous fat. Now it makes sense. Now it makes sense. So now this epidermis, I am looking more, going more inside. See the beauty of God. Now I am going more inside towards looking what is present in between this. So this is this, or this is this. Are you able to understand? So the layers, the epidermis has further layers. First thing, before going into the layers of epidermis, please, before going into the layers of epidermis, we want to have an orientation study in orientation, because people get confused in skin. So we will write, I want to show you a diagram. I want to make a diagram for you so that you get a clear concept. So outside is epidermis, outside is epidermis. Okay, sir, this is epidermis. Very good. Sir, after epidermis comes the dermis. Very nice. Sir, epidermis comes the dermis. Very nice. But sir, there is a point, a junction where I am able to appreciate that epidermis is coming in contact with the dermis. Epidermis is coming in contact with the dermis. That place is called dermoepidermal junction, DEJ. That place is called dermoepidermal junction. Dermoepidermal junction. The concept should be clear. If someone of you wants to become a dermatologist, if you don't have basic on the first year of dermatology residency, first day of your dermatology class, you will be learning again this topic. If you learn it here in the first year only, things become very easy. In the pathology also, it will become easy for you to understand what is a papule, what is a macule, what is a blister, what is a pemphigus. Okay, it's a acanthocyte. So dermoepidermal junction is the point where the dermis and the epidermis. Okay, this is a place, a junction. What is it made up of? Dermoepidermal junction is made up of, let me write the name first, dermoepidermal junction. Okay, DEJ. So it is made up of CT. What is CT? Connective tissue. It is made up of connective tissue. Okay, understood? So this is all the connective tissue membrane. Okay, so now what is this? What comes? What comes in this is the fat globule. Third, fat globule, and it has fibrous septations in between. It has fibrous septations. Let me draw the fibrous septations. Septations. So it has fibrous septations in between these fats. These fats have fibrous septations. Okay. So this is what is total, you see as a, if you see the total architecture of skin. Okay, easy. Now, skin, before going into epidermis, I want to tell you, skin is not that it is just this thing, these things are present. You also have presence of, appendages. You have hair, isn't it? You have nails, you have sweat glands. Okay. So what are appendages, by the way? What are appendages? Appendages means, appendages you can call to what things you can call appendages. Uh, so you can call, I think, nails. These are appendages of the skin. Sweat glands. These are appendages of the skin. Pilosebaceous unit or pilosebaceous unit, whatever you call. Pilosebaceous unit. Pilo means hair. Pilo means hair. So these all are included in the appendages part of the. Okay. So, oh, by the way, what are the sweat glands? We have two types of sweat glands: apocrine, ecrine, isn't it? Apocrine, ecrine. So apocrine, where are the apocrine present? Apocrine are present in the axilla, isn't it? In the areola of the nipple of the female, isn't it? No, areola in the breast, and perineum also. You have apocrine. So apocrine are present in the axilla. Apocrine glands are present in the perineum. They are present in the areola. So apocrine unit will open. The apocrine unit, your apocrine sweat gland will open into the pilosebaceous unit. Remember, apocrine will open into pilosebaceous unit. Okay, but eccrine gland will directly open on the skin surface. So should I draw it like this? Should I draw the diagram like this? If this is the same epidermis and I have a pillow hair follicle, if I say I have a hair follicle like this and a gland is associated with it, this is a gland. So can I say this is an ecrine? Can I say this is an ecrine? Can I say this is an ecrine? No, isn't it? Ecrine will directly open into the skin. Okay. So associated gland with the pilosebaceous unit will be apocrine gland. Okay. But eccrine gland will open like this directly. Eccrine gland will directly open on the skin surface. Okay. Now if I take these were the things I had to tell you. You know, if I have to show you now this epidermis, if I talk about this epidermis now, this epidermis, it has layers, further layers. Those are shown by this diagram. See, this is all. Can you see, appreciate? Can you appreciate? Let me delete this. I have to focus here. So can you appreciate that here is the junction? What is this junction? Epidermis is this much, isn't it? This all is epidermis. This all is epidermis. Are you able to understand, my dears? There was some problem, I think, suddenly because it's raining and maybe suddenly it stops. My mood gets, I have a flow in my brain, but then I have to again. Okay, let us start. Okay, so this all is epidermis. This all is epidermis. And in my first year of MBBS, I had a difficulty remembering things because I did not had any concept of mnemonics at that time. While preparing for the entrance examination, I knew there were some mnemonics I have to make, but did not knew that I had to learn mnemonics. I used to learn it in a crude way: corneum, lucidum, granulosum, spinosum, basal. This is how I learned. Okay, hitting my head to the wall. But you have to learn it in a simple, giving you some mnemonics. Okay, how to learn all the layers. So come, let's get famous mnemonic. Let's get some. Let's get some. Come, let's get. But you should make a mnemonic in such a way which works. Anyone can make mnemonic, but they are all fizul and bakwas mnemonics. Okay, we have to remember them on the exam day, all the 19 subjects. So that's why I take this mnemonic so that you can remember. Okay, come, let's get some. What? Okay, let's get some bread. Okay, so stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basal. Okay, so you have to go from outside to inside. Okay, this is the way you have to remember. How will you understand? Yeah. Okay, yes. So epidermis, if I talk now general, this is the layers you have to remember: stratum corneum, stratum lucidum, stratum granulosum, spinosum, and basal. And after that you will see here the dermis, and this is the derm epidermal junction. Now epidermis, talking about the epidermis, the epidermis is more thick. It is more thick. It is more thick. on your palms and soles. Palms and soles. It is more thick on the palms and so approximately I think 1.5 millimeters. So thinnest on your eyelids and thickest on your palms and soles. Okay. So a question has been asked a long time back about the epidermis. So where is the epidermis? So ectoderm is derived from ectoderm. But dermis is not derived from ectoderm. Dermis is derived from mesenchyme. I have taught you. Mesenchyme. Dermis is derived from mesenchyme. Ectoderm is epidermis. Dermis is mesenchyme. What cells do we see in the epidermis? So write the cells, please. So the cells which we see in the epidermis is keratinocytes, melanocytes, melanocytes, Langerhans cells, Merkel cells. Okay, not the Chancellor of Germany, Angela Merkel. So this is Merkel cells. Okay, so so if I go from bottom to top, so what is the arrangement? It is stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, stratum corneum. This is the way we are going from bottom to top. Okay, and sir, sometimes in the books you will be seeing that they are saying that four layers are present. They won't be writing about lucidum. They won't be writing about lucidum. They will be saying four layers. There is a reason. Books will be writing there are four layers. Okay, we're telling you later why. So let us share it here. Okay, so see, now what is, we have to individually talk about the layers. So if I talk about the stratum basal, this stratum basal is basal layer. This is the basal layer. Okay, this layer is actually the dividing layer. Okay, this layer is actually the dividing layer. And which type of cells does it contain? Columnar cells. Cells. Columnar cells. Okay. If I talk about stratum spinosum, I should write in short, that is stratum spinosum. That is, it is called as prickle layer. Why? It has some spiny arrangement of cells. Okay. So prickle layer. If you want to see desmosomes, do you remember the junctions, desmosomes? If you want to see desmosomes very nicely, you should go to the stratum spinosum layer. You should go to the stratum spinosum layer. See, it has again stopped. I don't know what's happening. It's not moving. Okay. So prickle layer. Okay. You want to see desmosomes, so more prominent desmosomes, more prominent desmosomes, more prominent desmosomes. Okay. Now these desmosomes are appearing to us like spines. Once we are seeing them under the microscope, there we can see these desmosomes are like a spines to us. So that's why we call it a prickle layer. Okay, put a star here. Put a star for the desmosomes. It's I think a recent AIMS question asked around 2 years back, I think. So this desmosomes. Okay, and this columnar cells. Okay, how we able to understand? Do you have any problem? So we are actually discussing one layer. Okay. Okay. So SG, then we go to SG, the granulosum. Okay. The granulosum, stratum granulosum, This stratum granulosum is granular layer. I'll be showing you later in the slides this. Okay, so granular layer. Why it's called granular layer? It has some granules. What are the granules called? Keratohyaline granules. Keratohyaline granules. It has some granules called keratohyaline granules. And we see some bodies. Please put a red pen. Use a red pen. Ordland bodies. Ordland bodies are found in these, in this layer, or you call it lamellar bodies. Lamellar bodies found in this layer. Okay, so you have keratohyaline granules and you have Ordland bodies. Okay, what is the function of this Ordland bodies? They will produce lipids. They produce lipids. Ordland bodies produce lipids. And then you throw it out. Keratinocytes have some channels. So you have keratinocytes coming out. So they will throw it out towards the outside. What is the basic function of this lipids which you formed? They are glue stick. What they are? They are glue stick. These lipids are glue stick. Glue. So these lipids are glue stick. What are they doing? They are attaching the keratinocytes. They are between the keratinocyte, one keratinocyte, one keratinocyte. So if you want to put a glue, give the award for these lipids. You have this glue. Should I tell you a disease in which this glue is absent, or in which this granular layer is absent? Okay, let us tell you from a dermatology point of view. So it is seen in psoriasis. So put a medicine. What is the clinical correlation for this? If you see what I am going to say, please listen to my words. This layer is absent in two diseases. Remember, two diseases. One is psoriasis and second is ichthyosis vulgaris. These are the two dermatological diseases which you see in your clinical practice. So main reason for this is your granular layer. Are you able to understand? Okay, easy, tasty, dermatology also. Okay, so let us go to the next layer, that is stratum corneum. Okay, stratum corneum. Okay, stratum corneum, can you see? We are now on the top, isn't it? We have from the medical school while learning. Now we are on the top of the success, isn't it? We are now doctors. So we have reached the top layer, this corneum layer. Okay, this corneum layer. I wish I get a cake like this. I am getting very much, attracted towards this diagram. I wish there is a cake with jam all around like this. Wow, so tasty. Yeah. On the birthday cake, I usually get the concept of the skin. Whenever I'm cutting a birthday cake of someone or I'm eating a cake, I can see the layers. Then I tell him that this is the corneum, this is the lucidum, this is the granulosum, this is the spinosum, if it has all the layers. Okay. Then I start talking about the psoriasis, ichthyosis vulgaris. Are you able to understand? So this stratum corneum is also called what? Horny layer. It's called horny layer. It's called the horny layer. So by the way, thickest layer. By the way, thickest layer. Can you see it is thickest layer or not? Can we see? Can we appreciate? Or we are not able to appreciate also because we are pink color blind, a new color blindness for you people. And some people, in the university also, when I start showing them on the slide on the projector that this is the thickest layer, they are still confused. Where is the thick? How is it thick? Now I have to teach them how to measure the thickness from the scale, isn't it? There are some varieties of people you may be tackling, very many varieties. So it is the thickest layer. Okay. By the way, this layer is dead. He is dead. Why dead, sir? And no nucleus. Can you see any nucleus? Can we appreciate any nucleus in this diagram? Can you appreciate any nucleus? Where is the nucleus, sir? No nucleus. Can you see the nucleus in the stratum corneum? Can we see in this region any nucleus? No nucleus. So that's why a dead layer. That's why a dead layer. Okay. But it is the beautiful layer. It forms the first line of defense of your body. That is the barrier function. It is actually preventing the entry of microbes. You have all that staph epidermidis on your skin, and even other floras or some displaced things can come inside. Microbials I am talking about. So they can come from the outside environment and settle inside the body. And also you also have fluids in your body, isn't it? You also have fluids in your body. So they should not be going to come out. They should not get leaked. So it's preventing that. I'm telling you the functions also. I'm not going to write. You have to write it by yourself. So it's actually preventing me. It is giving us a barrier function. Okay. I told you some time back, I told you that you will be tackling in the books, there will be written only 4 layers in some books. So why they are not writing about lucidum? According to lucidum, actually, lucidum is also only present in your palms and soles. The lucidum layer of epidermis is present only in the palms and soles. Okay, because it contains some degenerated keratinocytes. Because it contains some degenerated keratinocytes. So remember, on your palms and soles, you have an extra layer that is lucidum. So is this section taken from the palms and soles or not? Is this section to study the skin taken from? Yeah, it has been taken from. So it has either been taken from a palm or a sole. Okay. Sometimes you may be tackling. So what you should write in the lucidum? Just one word. Just one word. Two words. No, sorry, two words. One. The first word is palms and soles. Second, degenerated keratinocytes. This is how you have to remember. It contains degenerated keratinocytes. Okay, easy, easy like. Next question. There will also be a name in your books that is the Malpighian layer. Okay, Malpighian layer. Malpighian layer. So this Malpighian layer is actually, that term is used for what? Your spinous and basal layers. Spinous and basal layers in combo. Okay. Okay. So should we talk something about these cells? Should we talk about these cells? So let us talk about the cells one by one. Little bit points. So let us talk about keratinocytes. This keratinocyte, why they are called keratinocyte? Because they contain a filament that is keratin. They contain a filament. Which filament, sir? Intermediate filament. They contain an intermediate filament called keratin. Basically, the filament, the function of the filament to provide you, strength. It provides you with strength. Okay, like you every time press your hands, put pressures on your hands, some mechanical pressures. So to prevent the, to give strength, you need a strength, isn't it? Once you are hitting, or you are doing this, or you are putting, things in your hands, you're doing everything. You have made buildings, you have, whatever, what not. Everything has been done by the human being, every activity. You have made large buildings, you have did, miracles in science or engineering, technology. So that is because of this keratin. Okay, that is because of this keratin provides the mechanical strength to your keratinocyte. Okay, don't go into that much detail, I'm just saying. So this keratinocyte is, this keratin is giving the strength to these keratinocyte. Okay, now, these keratinocytes are making layers. Okay, they are making layers. Now I tell you one thing. There is a term used in the dermatology, I think, that is the turnover time. There is a, you have to read it in dermatology, but I am giving an association here. Okay, so turnover time or transit time. There is a concept in keratinocytes. What is this time? The time taken for a basal cell to reach, because you all have to shed, isn't it? The dead, all things are going up. So the highest deadest deadliest or the highest dead part of the skin is in the corneum, isn't it? If you want to find the dead cell, final dead cell, it should be on the corneum. So things should be coming towards up, isn't it? Things should be coming towards the corneum, stratum corneum. So the time taken from the basal cell, these basal cells to reach to the skin surface, that time is called transit time.